Hello everyone, myself Lokeshwari. In this video, we are going to see about bacterial wilt of potato. Bacterial wilt of potato is otherwise known as brown rot. This is one of the most destructive diseases of a potato, and the disease has been estimated to affect about 3.7 million acres in approximately 80 countries worldwide. This disease is known to occur worldwide in the wet tropics, subtropics, and some temperate regions. This disease is caused by a soil bacterium Ralstonia solanaceum, which affects 200 plant species in 50 plant families. This is one of the most damaging plant pathogen. The strains of this pathogen affect more than 200 plant species. The strains of Ralstonia solanaceum have been conventionally classified as races and biowards. Brown rot of potato is classified either by race 1 or race 3 strains of Ralstonia solanaceum. In the United States, race 1 is endemic and can cause bacterial wilt on several major crops such as eggplant, pepper, potato, tobacco, and tomato. Potato wilt bacterium mainly inhabits the roots and enters the root system and then soil pest. On potato, bacterial wilt otherwise known as brown rot, southern wilt, sore eye, and jammy eye. Host plants. Bacterial wilt attack more than 200 species. Economically important hosts include banana, beans, eggplant, peanut, pepper, tomato, tobacco, and potato. Two common weed hosts that have been attacked by the disease are nightshade and thorn apple. Environmental condition. Bacterial wilt of potato is generally favored by the temperatures between 25 degrees Celsius to 37 degrees Celsius. It usually does not cause problem in the areas where the soil temperature is below 15 degrees Celsius. Under the condition of optimum temperature, infection is favored by the wetness of the soil. Once the infection has occurred, symptoms will often be secured with hot and dry condition, which has wilting. Spread of the disease. Irrigation water and flood waters, contaminated soil, nematodes, infected equipment such as seed cutters, second handbags or half-ton bins, seed dissemination. The wilt bacterium can survive for periods up to 2 to 3 years in barrow fields. Symptoms. There are 7 symptoms that causes the bacterial wilt of potato. They are: first, symptom on brown rot of potato caused by the Ralstonia solanaceum showing wilting of youngest leaves on plant. Second, symptoms of brown rot of potato caused by Ralstonia solanaceum showing wilting and stunning of the plant. Third, brown discoloration of the stem tissues that is caused by Ralstonia solanaceum. Fourth, grey-brown discolorization of vascular tissues and bacterial ooze in plant tuber infected by the Ralstonia solanaceum. Fifth, bacterial ooze from freshly cut section of a geranium stem infected by Ralstonia solanaceum. Sixth, bacterial ooze exuding from the eye of potato tuber infected by Ralstonia solanaceum. Finally, bacterial streaming in clear water from the stem cross-section of a plant infected by Ralstonia solanaceum. First, wilting of youngest leaves of the plant. At the early stage of the disease, the first visible symptom of brown rot are usually on the foliage of the plant. These symptoms consist of wilting of youngest leaves at the end of the branches during the hottest part of the day. Second, wilting and stunning of the plant. At this stage, only one or half of the leaflet may wilt and the plants may appear to recover at night when the temperatures are cooler. As the disease develops under favorable condition, all the leaves in a hill may wilt quickly and desiccate although the dry leaves remain green, leading to the general wilting and yellowing of foliage and eventually plant death. Another common symptom that can be associated with the brown rot in the field is stunting of plant. These symptoms may appear at any stage of the plant growth. Third, brown discolorization of stem tissue. In young potato stems, infected vascular bundles may become visible as long, narrow, and dark brown streaks. Succulent plants of highly susceptible varieties collapse the stem and may also be observed. Cross section of the stems and stolon may reveal brown discoloration of the infected tissue. Fourth, grey-brown discoloration of vascular tissues and bacterial ooze in potato tuber. Brown rot symptoms may be present in potato tubers at the later stages of the disease. Cross section of the infected potato tuber may reveal a grey brown discoloration of the vascular tissue. It is also called as the vascular ring. As the infection progresses, the discoloration may extend into a pith or cortex of the tuber. A milky white sticky ooze, which indicates the presence of bacterial cells, might also be observed from the fleshy cut section of the infected tuber. Fifth, bacterial ooze. The symptom expression is favored by the high temperature between 85 to 95 degree Fahrenheit, and the symptom of the disease may progress rapidly after the infection. In the favorable condition, symptoms less plant may remain latently infected for extended period of time. After the infection, the pathogen may survive in and be spread from the infected plant. A common sign of brown rot observed at the surface of the freshly cut section from severely infected stem is sticky milk white exudate, which indicates the presence of dense masses of bacterial cell in the infected vascular bundle and particularly in the xylem. Sixth, bacterial ooze exuding from the eye of potato tuber. Bacterial ooze may be visible at the eyes or at the point where the stolon attached to the tuber. These signs may not be visible early in the disease development. 7. Bacterial steaming in clear water from stem cross section of a plant. Another common sign of the disease is observed when the stem cut section are placed in the clear water. It consists of a viscous white spontaneous slime streaming from the cut end of the stem. This streaming represents the bacterial ooze exuding from the cut ends of the colonized vascular bundle. Causative organism. The most causative organism that causes the bacterial wilt of potato is the Ralstonia solanaceum species. It is a gram-negative rod-shaped sticky aerobic bacterium that is 0.5 to 0.7 to 1.5 to 2 μm in size. It has a one-polar flagellum. The agar colonies are small, roundish, smooth, wet, shining, become brown rough formation of black water-soluble pigment. It is very sensitive to desiccation and is inhibited in culture by low concentration 2% of sodium chloride. The optimum growth temperature is 82 to 90 degree Fahrenheit. However, some strains have a low optimum temperature of 80.5 degree Fahrenheit. Ralstonia solanaceum is widespread in tropics and subtropics around the world. These strains were initially subdivided into races and biovars. So far, 5 races and 5 biovars have been identified within the species. Both race 1 and race 3 can cause brown rot of potato with similar disease symptoms. Race 1 corresponds to the biovars 1, 3 and 4 that includes the member of all 4 phylotypes. Epidemiology of the disease. Ralstonia solanaceum is a soil-borne and water-borne pathogen, the bacterium that can survive and disperse for various periods in the infested soil or water that can form a reservoir source of inoculum. In potato, the brown rot pathogen is commonly found as tuber-borne. Bacterial wilt is one of the most destructive diseases of the potato. The disease can cause total loss of a crop and prevent the use of land for potato production in several years. The life cycle of the Ralstonia solanaceum is the bacterium usually infect the potato through the root and then exposed to the healthy plant in the field. Wilt symptom initiation and Ralstonia solanaceum multiplication under favorable condition will happen. Then the systemic infection caused typical bacterial wilt symptoms led to the plant death. The disease spread through irrigation water, rain splash, natural wounds in the root hairs, root tips, root to root contact, nematode injury and the improper culture practices. Then the Ralstonia solanaceum spreads to the entire field and can cause the wilt outbreak. The saprophytic survival in the soil and the weed host. Then the Ralstonia species return to the soil environment from infected planting material and provide an potential inoculum source. Brief explanation of life cycle of bacterial wilt of potato. This bacterium usually infects the potato plants through the roots. Wounds are at the point of emergence of lateral roots. Then it enters the potato plant through the root system. Entry via wounds or natural root openings such as cracks, root tips, or root hairs. Ralstonia solanaceum can also infect through infected seed tubers or contaminated irrigation water. It migrates through the plant vascular system xylem and begins to multiply rapidly within the xylem that absorbs the water and nutrient flow. This bacterium spreads through the plant's vascular system, colonizing the stem, leaves, and eventually the tubers. During this phase, the bacterial population increases significantly within the plant and shows wilting. Ralstonia solanaceum can invert the developing tuber, causing internal brown discoloration and rot. Infected tubers may exhibit vascular browning near the eyes and rotting as the disease. As the infected plant tissue decomposes, it releases into the soil and then spreads to the nearby healthy plants through contaminated soil, water, farming equipment, and infected tubers. It can also be transmitted via nematodes or soil-borne insects that creates wounds in the plant disease. Then the cycle continues as the Ralstonia solanaceum survives in the soil, water, and crop debris until it finds a new home. Diagnosis and identification of bacterial wilt of potato. Symptom identification is the first step for the early diagnosis of brown rot of potato. Ralstonia solanaceum is either symptomatic or asymptomatic. They cannot be used to identify the race or biowar of the organism. Screening tests include stem streaming, plating on semi-selective medium, immunodiagnosis assay, nucleic acid based identification. Several rapid screening tests such as immunostrips are available commercially for the rapid and field detection of Ralstonia solanaceum. Biochemical test is also used for the bioware determination of Ralstonia solanaceum. This is based on the differential ability of the strains of the pathogen to differentially produce acids from several carbohydrate sources including disaccharides and sugar alcohol. Several identification of the strains of Ralstonia solanaceum can be assessed with the several nucleic acid based methods such as DNA probe hybridization and especially polymerase chain reaction amplification with specific probes and primers. Race determination is not generally possible because Ralstonia solanaceum strains usually have numerous hosts and do not have a race cultivator specificity on plant host. Prevention and control of bacterial wilt of potato. Use disease-free tubers, use the certified soil and avoid infested soil. Plant that only are certified disease-free seed potatoes to prevent introducing the pathogen to the new fields. Sulfur limestone is used for the treatment. Field sanitization. Remove and destroy the infected plant and crop residues to minimize the source of infection. Crop rotation. Used to rotate the potatoes with non-host crops, example cereals, legumes, for at least 3 to 5 years to reduce the soil population of Ralstonia solanaceum. Thank you.